introduce our next speaker, who's Jean-Philippe Vert. I think that many of you in the community know him. He's an honored member of the bioinformatics French community. He has a prestigious pedigree. He studied at many prestigious higher education institution, École Polytechnique. He graduated from École des Mines. He did his PhD at Université Paris 6. He studied in, he did research in France, the United States, and Japan. And his research topic is devoted to deep learning and computational biology. Many of the not so young audience of the community probably had the chance to listen to some of his lectures. I hope that some of the newer members of the community will have the opportunity in the future, because it was each time a very nice experience. And today his presentation is concerned with deep embedding of biological sequences. So Jean-Philippe, please astonish us once again today.
Thank you very much for the kind introduction, and thanks all organizers for organizing this meeting, especially in these complicated times where organization can be more more complicated than usual, and also for giving me this opportunity to speak today. It's a really great honor. So I would like to discuss today. I will talk about my own, some of my own research project, but the broader picture of what I'd like to say is suggest that maybe in the field of biological sequence analysis, which historically has been one of the most important topics in JOBIM in particular, but more generally in bioinformatics. So I'd like to suggest that maybe in that field there's a big change happening now, and so share with you some of my ideas on that change and whether how much it was pursuing or not. So the change I'm talking about is really, it's not a surprise, by the way. There will be no surprise in the talk, but maybe for some of you who are less familiar with, I will detail a bit what I mean with the change. So the change is inspired by what happens in similar fields. In particular, you hear of machine learning, deep learning everywhere. There's a field that has been a lot impacted in recent years, which is natural language processing. So by we typically, NLP means natural language processing. So it's the field where we develop algorithms to analyze language, but human language like French, English, Russian, Chinese, etc. So NLP is not a new field. There's a long tradition of a variety of ways to analyze text. So here I'm mostly talking of written texts. But it's fair to say that in the last 5 years or so, the techniques used in the field of NLP have changed a lot, in particular with the advent of deep learning. And something that has been found in particular very useful is that, instead of developing specific algorithms and models for specific tasks and specific languages, and for example, it used to be that to detect verbs or grammar or understand the structure of a sentence in some language, some people would develop specific algorithms. Nowadays, it seems that the magical formula that came with deep learning is somehow to take lots of data of any text, could be of good or bad quality in any language, and train models where all the text is mapped or represented as vector in some space. So vector is just a series of numbers. And somehow the techniques used in many cases find a unique way to map any text into a vector in such a way that this vector representation is a good starting point to solve many tasks in parallel. And by many tasks, I mean the same embedding or the same vector representation is used to solve tasks like machine translation or text generation, text correction, speech recognition, etc. Right, so this is really what, this is one way to say what deep learning is about in that field. It's about learning to embed sequences or text into a vector, and then from the vector representation, do interesting things. And this has led to remarkable progress, like accuracy has really gone up in, for example, translation quality. It's implemented nowadays in, on the web, in your smartphone, in many applications. And so the whole field of NLP, I think, has been dramatically impacted by this kind of new way to process strings, so to process sequences, map them to vectors. So the devil is in the details, of course, right? What's important is to figure out how you transform a sequence into some vector representation, and then how from the vector representation you solve machine translation or text generation tasks. But there is this bottleneck at least, which is, and this central idea, which I find quite fascinating, to map your data to vectors and then use the power of basically linear algebra and matrix multiplication in that space to do fancy things. And by the way, I talk of NLP, but exactly the same thing has happened in computer vision, where in that case, images are mapped to vectors. We hear a lot about graph neural networks, for example, so how to represent molecules or larger graphs, again, as vectors in order to solve tasks. So this is quite a central idea, and the power of deep learning is to provide tools to parameterize, if you want, the model that transforms the input data into a vector, and then from this vector does something useful, and provides great tools in order to optimize the parameters by gradient descent, basically. Right. So I will not go to the detail of what each component is, but there is this idea to use vector representation. And so basically what I would like to suggest today or to discuss is whether this idea is not completely new, of course, in bioinformatics, but whether we could push it really to further in bioinformatics. And so asking the question, if we move from natural language processing to, I call that in this talk BLP, so biological language processing, meaning if we want to analyze text written in the language of DNA or proteins, would it make sense to try to systematically find, just like in NLP, representations of these sequences as vectors? So here I plot the vectors as dot because as soon as you have a vector, you have coordinates. So it's the same to talk of a sequence of numbers or of a point in some feature space. And so would it make sense to try to embed all these sequences as vectors, so in some continuous space, and from there try to solve a variety of tasks, which in the case of BLP would not be machine translation or speech recognition, but which could be protein annotation or DNA annotation, classification, search. Search engines typically are based on embeddings as well, automatic generation of sequences with some properties, etc., etc. Why? So this is a question I like to ask, and I'm not sure about the answer. I think many people are trying to work on that these days with very promising results, and I see that a bit as a game changer in bioinformatics, in the sense that if you take a textbook of sequence analysis, maybe the tools that are in the textbooks in bioinformatics will not be useful anymore in a few years if the default method is about this learning to embed sequences and then doing linear algebra to solve search or classification or generation tasks. Okay, so as I said, this is not a new idea, and actually, since there is a long tradition of neural networks in bioinformatics. Then there's been, kernels, etc. So all of these methods were related. But the central idea to push, to follow NLP development, deep learning, and try to map sequences to vectors and then do something useful has been pushed by many groups in recent years. On this slide, I just mentioned, and I will not detail them, but mention, a tiny fraction of papers that were published recently in that domain. Notice that this is, many of them are in very high impact journals. And many of them are about borrowing techniques from NLP, so algorithms, deep learning models for sequences that work for English. You can just take them and try to use them on proteins. And for example, this year there was a nice paper in PNAS. The biological structure and function emerged from scaling unsupervised learning over 250 million protein sequences. That's just one paper that very nice paper that says, well, we have so many protein sequences. What if we tried to train a model just like we do when we have hundreds of millions of texts in English, which are used in, in Google, Facebook, etc. If you applied the same model on proteins, what do we see? And they study that and show that once you learn some embedding of the proteins, then you can see how many other things that we know about proteins are captured by the embedding in the continuous space, so about the structure of proteins, the function of proteins, etc. And so these are all, promising results showing that there's potential in a kind of unsupervised way, because here the embedding part can be trained somehow without label on the sequences. So it's possible to train this model to learn embeddings that start to capture biology there. All right. So after this introduction, I'd like now to detail some of the works we did with my colleagues and students, and to focus on 3 topics, which have something in common, which is that I believe that BLP, so biology, can be inspired by natural language processing, but that is still different. And so the topics I'll discuss today are exactly about the differences. Like, suppose we want to get inspired by the techniques using NLP. So we want to borrow NLP techniques to BLP. Are there differences? What are differences, and what should we change, or what should we try to change if we want to take the most out of protein or DNA sequences as opposed to English or Chinese sequences? And so I'll discuss 3 things today, and this is by no means a complete list of the differences, but these are things which are different and which we worked on. So this is to illustrate a bit the kind of research questions that can be addressed when you think about BLP in particular. So the first thing I will discuss is how to decide what's the dictionary or the alphabet when you work with biological sequences. Second topic will be how do you incorporate prior knowledge about symmetries, and in particular when you study DNA. For some applications, there is the classical reverse complementary symmetry that exists, and that does not exist in NLP. So this is a difference, and maybe this requires adapting the models used in NLP. And finally, I'll talk about the problem of sequence alignment, which does not really exist in NLP, but it's central in biology. Maybe that's the first kind of algorithms you learn when you start studying bioinformatics. It's how to align sequences. So can the alignment problem be solved by these embedding techniques, and can it be useful as well? Okay, so I will discuss these 3 topics in turn, and then it should be time to conclude when I'm done. Okay, so the first topic is what should the tokens be in NLP? And to be precise, this requires some. So when you study what models are used in NLP, so the link between NLP and BLP is that you study sequences, but there's always something that that needs to be done. Is when you start from a sequence and want to build a vector out of it. So all the deep learning model transforms sequences into either one vector or a sequence of vectors. Then there's always a step at the beginning, at least, that decomposes the sequence, written in ASCII or in some language, into a sequence of tokens. And it's not super clear what tokens are, but typically in NLP, tokens are created by taking your raw sequence of letters and merging together sequences of letters to form either words or subwords. And it's not easy, by the way, even in NLP, when you study when people develop models for English, there is a consensus on how to do, but it's not a completely closed question. But so on this picture, you see that, for example, you can decide to work at the level of letters on the left. So some models are based on characters. These are not the ones that work best, actually. These are not the ones that are implemented for tools doing automated translation. Another extreme would be to work at the level of words. So here, one difficulty is how do you define words? You can say a word is just when you have a sequence of letters separated by a space or punctuation. But again, this one is not the best representation, in particular because many words are almost the same and mean something similar. And if you define them differently, they correspond to 2 different concepts, in the embedding space. So it has been found in NLP that what works best, at least for many applications, is to automatically learn a way to what's called tokenize, meaning transform a sequence of characters into a sequence of bigger chunks called tokens, which typically try to capture some semantic of the words. And so this would be like the first step before then trying to, learning embeddings for each token. Then you have a sequence of tokens becomes a sequence of vectors, and then you have several layers of processing to process the sequence of vectors. Right, so there is some work that and super important. Like you see differences in NLP, whether you work on characters, on words, on subwords, etc. This has impact on the performance at the end. So an open question is, if you want to borrow these techniques for proteins or DNA, what are the tokens? How do you tokenize that? And it's very clear that one option is to work at the level of characters. Like you could say, well, a sequence ATTATG, blah, 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 is first an A, then a T, etc. So this would correspond to, in NLP, the character-based models. But for example, it's known in NLP that this is not what gives the best results, because there is semantic by aggregating characters together. And I think in biology, it's fair to say that we also know that there are motifs or there are sequences of letters that typically have some, can be considered as building blocks of sequences. So it may make sense to try to not to see the sequence as a sequence of letters, but to try to decide what would be a good alphabet or tokenization process to first transform this DNA sequence into a sequence of tokens that then would be the first layer of some embedding in a continuous space. All right, so I'll discuss, some ways to tokenize and see a bit where we can go from there. So a classical way to tokenize, in fact, in DNA. I mean, people have always, I've always thought about this problem, and there are many ways, but one of them is to tokenize a sequence as a sequence of k-mers. A k-mers being just a sequence of k letters. So this means that when you have a sequence of DNA, like the one shown on this slide, you can, instead of seeing it as a sequence of letters, you can see it as a sequence of triplets of letters. Like in this case, you start from the left. The first token is AAG, because these are the three first letters of the DNA sequence. Then you move by one position, and the second token in this case would be AGC. Right, so these are overlapping k-mers. And so there is no loss of information, right, between the seeing the DNA sequence as a sequence of letters, nucleotides, or a sequence of triplets of nucleotides. But in a sense, for machine learning models, choosing the sequence of triplets of nucleotides may be better adapted if we believe that this is where there is some information to capture in the sequence of nucleotides that can be captured by embedding these as a token. Right, so if you go that way, then it's possible very quickly, to say, well, there is one simple way to embed a sequence as a vector. It's just to extract, to see it as a sequence of k-mers. And for example, if, without going to deep learning yet, but just being shallow learning, so just saying I replace my sequence by a vector representation, and then I do machine learning on that, you can just count the occurrence of each k-mer to transform a sequence into a vector, which is shown on this slide. So if you choose k as the length of the k-mer, you can imagine that you have 4 to the k dimensions if you work on DNA with 4 nucleotides. And so you can represent a sequence of DNA by a 4 to the k dimensional vector. So 4 to the k gets large very quickly with k. That is indexed by all possible k-mers and just counts. So for each k-mer, the number in the vector is how many times the k-mer occurs in the sequence. All right, so this is one embedding of a biological sequence as a vector. And from this vector embedding, you may want to solve many tasks in biology. It's not very powerful, we'll see, but that's a good beginning. So actually, that's something we did with colleagues, in particular Kevin Verviers, who is now, I don't remember, in UK, I think, and Pierre Maillet and others from Biomarrieux. So we worked on that a few years ago to solve problems of metagenomics sequence classification. So here, that's a problem where we want, metagenomics is about sequencing some environments, collecting lots of short DNA sequences, and then you want to learn something about the environment. And one possible way to run something is for each DNA read that has been sequenced to try to predict which species it comes from, right? You have a database of possible species, organisms, so typically bacteria. And when you sequence an organism, you would like to identify what are the bacteria in the environment. So this can be formulated as a machine learning problem where as input you have a DNA sequence and as output you have a species, right? So it's a multi-class classification problem. And on that work, we propose to use machine learning to do that instead of other techniques like alignment of sequences to reference genomes. And so if we use this spectral embedding technique, then each DNA sequence gets represented as a 4-to-decay dimensional vector, the one I showed before. And then when you want to transform this vector to a prediction, you need to learn a model. If you think of the simplest possible model, which would be a linear model, then you need a number of parameters, which typically is equal to the size of the vector, so 4-to-decay, multiplied by the size of the classes, because for each class you need a linear model. So you end up with 4-to-decay times number of class parameters. So this works, and in the paper we show that it works pretty in a competitive way compared to other approaches. But one bottleneck here is that the number of parameters gets large. So in memory of your computer, this requires big models. As soon as k gets larger, and so for example in that paper we could only go to k equals 12 because we worked on big computers with I think it was something like 64 giga of RAM at the time. But the thing is, coming back to the question of whether using k-mers is useful or not, in that paper, at least we demonstrated that the performance increases with k, right? So it means that there is more information in the sequence of k-mers than in the sequence of letters, in particular because we just count them. And we would like to ideally to go to larger k because we observed that in our experiments, k equal 12 was the best performance, but we couldn't go to 13 because we didn't have enough memory. All right, so a few years later, I worked with Romain Ménégo, who just got his PhD recently, on some extension of that model to try to solve the problem of trying to go to larger k. And here we use techniques which get a bit closer to the NLP approach. So Romain developed something called FastDNA, which is inspired by actually a model called FastText in NLP. Which works as follows. So it's a bit the same idea as a k-mer. So you start with the DNA sequence. When you have a DNA sequence, you extract the k-mers it contains. Sorry, I lost my screen, so I need to retype my password. Sorry for that. Yeah, so you extract a sequence of k-mers. And then instead of just making a big vector to count how many k-mers there are in the sequence, you can...
I'm sorry to interrupt, but I don't think you have your screen anymore.
Ah, okay, sorry.
Yeah, your screen is not shared anymore. And Mahendra, maybe we can take the opportunity. Someone is raising the hand, so maybe you can ask her why. Can you see the people who are raising the hand?
Nope.
If you click on participants,
okay.
You have a list of people, and on the top you have the spectators, and people are raising their hand. But I don't know if it's
maybe it's because of the slides.
Maybe it's because of the slides. I don't know.
So do you see the slides now?
Yeah.
Okay, sorry for that. Just my computer turned to sleep, so I restarted it, but a lot of slides. So okay, so continuing on FastDNA. So FastDNA is an adaptation to DNA of a model which is used for English called FastText, developed by Facebook Research. And so the idea of FastDNA is to extract k-mers, but then each k-mer is represented by a d-dimensional vector where d is much smaller than the number of k-mers. So there is a compression of k-mers, and this is like these are free parameters to be optimized, right? So the model will optimize these parameters, but it goes through this compressed representation for each k-mer. And then when a DNA sequence as a sequence of k-mers, then the embedding, so the representation of the DNA sequence itself, would be the sum of the k-mers embeddings. right? So this goes, this now we are closer a bit to what people call deep learning, in a sense that we learn a representation, we learn the k-mer embeddings that then allow to represent a sequence as a vector, and then from the compressed vector representation for the DNA sequence, we can then again make, for example, linear models to predict the class of the input DNA sequence when we want. So we apply that to the same metagenomics problem. Right, so if you look at the graph on the bottom left of this slide, it somehow summarizes what we do here. Instead of directly connecting the k-mers to the number of classes, we go through this bottleneck layer. And so this implies, in terms of memory requirements, that the number of free parameters of this model, which are optimized during machine learning training, is reduced, because you just need on the left of the bottleneck to store a matrix of embedding for each k-mer. So this one has size, there are 430k k-mers, which are presented in d dimensions. So the number of parameters is 430k times d. And then once you have d dimensions and you learn linear models to predict n classes, then the number of parameters is just d times n. So this can be much smaller than the total 430k times n classes that we had before. Romain worked on that, implemented that, and showed that it's quite good, in a sense that I will not go to the details of the results, but long story short, it works better than the previous model where we just did k-mers. So the fact that we can compress the k-mers is useful, in particular because it allows us to go to a higher case. Not really much, because we again, we need an bottleneck dimension large enough. Typically, we need at least 50 or 100 dimensions in the bottleneck layer, in the embedding dimension, and the growth in k is exponential. So on that paper, at least with the normal computers, we could go to k equals 14 instead of 12. But going from 12 to 14 already brought some significant improvement and robustness to noise that compared to what we had before. All right, so then we continue working on that, and motivated again by the question of, can we increase k further? And here an idea that Romain proposed was to say that if the bottleneck, so if the problem with the memory is that when we have k-mers and we need to learn representations for them, so learn embeddings, meaning learn a vector for each k-mer, then we have to store this big matrix where you have number of k-mers times d dimensions to store somewhere. And this thing can get big. So here we talk of 50 gigabytes of RAM for just k equal 14 and 50 dimensions. So if you want to go to k equal 20 or 30, then you multiply this number by 4 to the 10 or 4 to the 20. So it's just impossible. And so an idea that Romain proposed was to say, well, in order to solve that problem, maybe you can cluster the k-mers together, and say that even though there are many different k-mers, maybe many of them should be represented by the same vector. So we should have a fixed number of vectors here that we can store in memory. And the question becomes how to cluster the space of k-mers when k is very large, then to attach each k-mer to representation, and then learn the representations of a group of k-mers. Right. And so this is what Romain investigated, and in particular, he used this idea of the Bruijn graph, which is a very classical concept in particular in bioinformatics, which is just a graph of k-mers. So it's a graph where the nodes are all possible k-mers, and where you put edges between two k-mers when they occur next to each other in one sequence, right? So importantly, the Bruijn graph depends on a data set of sequences, and it's a way to represent a data set of sequences as a graph. So, for example, here you have two sequences, two short sequences, and the corresponding Bruijn graph. So imagine if you have a data set of genomes, then you can build again a bigger Bruijn graph that would encode all the genomes as a path in this graph of k-mers. And so if you think of, if you want to train a model, for example, on metagenomics on a training set of DNA reads, then it's easy to think that if you make the De Bruijn graph of your training set, and if you have sequences of k-mers in the De Bruijn graph which are next to each other but without connection to other k-mers, this means that these k-mers always occur together or not together in the sequences, and so in the sense they are indistinguishable from a machine learning viewpoint, like in this case. You see that you have CAG and AGG, which are two k-mers consecutive to each other, and they are either both together in sequences or they are absent together. So this means that there is no discriminative difference information between the two k-mers. And so this means that probably you don't lose any information if instead of learning two representations for these two k-mers, you compress them to learn a single representation for these two k-mers. Right, so based on this, if you fill up this ID, then the next step is to say, well, if you start by building the De Bruijn graph, then you can extract all linear parts of this graph, which are called sometimes contigs. So you create the compacted De Bruijn graph by merging together the sequences of k-mers which follow each other without branching, and then you could say, well, instead of learning k-mer representation, I will learn a representation of these bigger boxes. Right, so the model, this leads to a slightly different architecture where to embed a DNA sequence, you again extract the k-mers. Then each k-mer you look in the compacted De Bruijn graph, the contig it comes from, and you learn an embedding for the contig. So you present a k-mer as the sum of the vector that represents the contigs it contains. And so the good thing with that is that it allows to go to much larger k. So for example, we tried with k equal 21, 25, 31. So the number of contigs does not explode. We observed that the performance was still increasing with k over a large range of k values. So this is really a net gain compared to the restrictions we had before due to memory constraints. However, there is a cost to that, which is that at some point, the De Bruijn graph depends on the training set of sequences. And so as soon as you have some noise in the sequence, or if you want to classify sequences which are really not really far away from your training set, then it may be the case that k-mers are in no contig, right? And therefore, there are more and more reads that you cannot predict in that model. So I think this is where we are now. I think there are many open questions, but it's just to show that this idea of learning embeddings leads to very promising experimental results, good precision, fast models, but that I think we are still a bit naive on how we tokenize the DNA sequences. K-mer, of course, is just a simple idea, so we try to push the limit of having larger k by playing a bit with compressing the representation using the Bruijn compact graph. But to me, there is the broader question of if we want to use embedding of discrete sequences, how do you first represent DNA or protein as a sequence of discrete tokens? All right. Second topic, and I'll be a bit faster for the next two topics. Don't be scared by how fast time goes. So second topic is a bit more specific to DNA, but I think it's interesting because again, this is something that's specific, so that does not exist in NLP, and it's studying what's the implication of the fact that DNA has this reverse complementary symmetry. So reverse complement, this is a word that I suppose most of you know. It just refers to the fact that DNA, so double-stranded DNA is you have two strands in DNA. So when we describe a DNA sequence as a sequence of nucleotide like A, T, G, C, blah, 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 this describes only one of the two strands. But if you are confronted with a machine learning problem that, where the input is a double-stranded DNA, then there are two equivalent sequences to represent it, right? You can represent it in one strand or the other. And so remember that reverse complement symmetry is about reversing from left to right to left the way you read the sequence, right? And in addition, changing A and T and changing C and G by symmetry to get the reverse complement sequence, right? So one double-stranded DNA sequence can be represented by two sequences of nucleotides. And so this raises the question that suppose you want to build a model that takes as input, a DNA sequence to represent a double-stranded DNA molecule. You would like, for example, if your model, for example, is given a DNA sequence predict class, like in metagenomics, I'm sure you would like your model to predict the same thing if you have one sequence or its reverse complement, because this describes the same molecule. It's just some arbitrary choice to decide that you show the strain 1 or the strain 2. So you want these invariants to reverse complement. There's something else that you may want, which is kind of illustrated on that slide. It's the question of translation symmetry, which is that suppose you have a molecule, double-stranded molecule of DNA presented on the bottom of that slide, and what you would like to predict, for example, is a profile. So it's a property along DNA, like you want to predict some epigenetic mark, or you want to predict the binding of transcription factors. So you can think that the ideal model that you want to solve is annotating a long double-stranded DNA with some signal on it. And to do that, you have access to short reads. So the short reads are illustrated here by these arrows, 2 on the right and 1 on the left, to illustrate which strand is sequenced. So ideally, what you would like is that your predictions should be coherent with each other, meaning that by design, if you show one sequence, a short DNA sequence to your model, it should predict the black profile on top of it. If you translate a bit your sequence to the right or to the left, then the prediction should be translated as well. And if you reverse complement, so if you show the same position either on the blue strand on the left or on the red strand on the right, then you would like your predictions to be the same, right? So these are like prior knowledge to tell you, depending on what you show as input to your model, what constraints you would like to have on the output. And my point here is just that if you don't do anything, this will not happen by default. Like if you take a deep learning model for NLP and you train it with sequences, there is no reason why it would know that it should predict the same thing, or symmetrize the thing or translate it, depending on whether or not you show a sequence or its reverse complement or small translation of it. So this raises one interesting question, which is, should we try to put this prior knowledge, so this property we want, somewhere to make sure the model we train will be smart enough to predict the same thing for one DNA sequence and its reverse complement, for example. And in fact, that's a question that a few teams have investigated recently. Here I mentioned four references. In particular, I mean, they all work on the same model, which is represented on the left here. I won't go to details, but think of it as a deep learning model. So you have layers. The bottom layer is just the input data. So it's a sequence of ACGT. Each layer computes a representation. There are arrows here and there to represent some constraints, on how each of these numbers is computed. So these are convolution layers, and there is some constraints on the weights, etc. But at the end of the day, it's a smart architecture that has a property that if you, as input, you show a DNA sequence, at the end you obtain a prediction. And if you show the reverse complement, then by design, on the top, you will obtain the symmetrized prediction. And similarly, if you show a translated version, you will get a translated version at the top, right? So it's an architecture that does not exist in natural language processing. It's specific to DNA and has the property that we want. Now, this has been proposed, and so one question we asked with my group is it possible to do different things? Like, are there other architectures which have the same property? Like, this one seems to work, but maybe there are other ones which would be worth having in mind, so that when someone wants to implement a deep learning model on DNA, she would have the choice of maybe this model or some other one. So can we characterize the set of models which have this nice property of being equivariant? And so here I will not go to any detail. I would just mention that we have recent tech report, and this is joint work with Vincent Mallet, who is a PhD student. In particular, I co-supervise him with colleagues at the Pasteur Institute. And so he studied specifically this question. So the question was clear, like, can you describe all operations, so all structures, all architectures of your networks that would be equivariant for DNA? And so equivariant is just a mathematical term to say that we want the model to behave well when we translate the input, or we reverse complement the input in such a way that the output will be translated or reverse complemented as well. And so I will not go to detail. There is a link to a paper, but I will just illustrate that it's possible to formulate it as a mathematical question. So to define exactly what we mean by being equivariant, you can guess it on that slide. And then we have a few theorems that basically tell us that it's possible to characterize exactly what operations you can do on the layer. So when we talk of translating and reverse complement, what does it mean when we have a multi-layer neural networks? This can be characterized mathematically. And in particular, we can see that we recover the previous, so the existing architecture, but there are many other architectures that could have the same property. Then in addition to that, Vincent was able to characterize all what's called linear layers. So if you have a representation that is equivariant, how you transform it in a linear way to some other equivalent representation. And the take-home message here is that convolution is all you need. So the only way to have a nice linear layer is to have what's called a convolution layer. Sorry for those of you who are not familiar with deep learning, but let's say it's just a standard thing in deep learning. And here the theorem tells us that that's all you can do, I mean, to be equivariant. So you need to have a convolution, and you need a convolution to respect some constraints which are written in these ugly equations. And in addition, Vincent was also able to show, the interest in deep neural networks often is to have nonlinear layers as well. And he was able to characterize what are the possible nonlinearities, and in particular showing that the one that exists is very nice because any nonlinearity is allowed. But if you try to extend and look at other representations which are equivariant, because we have all of them, then depending on the ones you choose, you can have different nonlinearities. All right, so I don't think the goal here is to detail the results, but just to, just wanted to tell you that as soon as you start asking a specific question, like, can we put this prior knowledge about DNA in the model? Then this can be formulated mathematically. We can have theorems. And at the end of the day, we can have a new set of tools to develop models to embed DNA sequence that respect some symmetries in DNA, which are specific to DNA and do not exist in even in proteins or even less in English. Very quickly, we tested these models on some benchmark experiments. So here the experiments are, you're given a DNA sequence and you want to predict DNA, so binding to DNA for a series of transcription factors. So you want to predict signal that can be experimentally tested by ChIP-seq data. And we observed that the existing model works well, so it's called RCPS. But when we extend and investigate newer architecture, which are allowed by our theorems, then it's not always very consistent, but we can have some better performance depending on the transcription factor, depending on the task. So it's not like there is a new architecture that works all the time, but there is a class of basic layers that can be used for double-stranded DNA. All right, I will conclude the talk quite quickly now with the last topic, which is about sequence alignment. No need to say that sequence alignment is crucial, and in biology, many people are, even in this audience, are much better than me on knowledge about sequence alignment. But this is something that does not exist in NLP. So I think an interesting question is, if we talk about embedding DNA and proteins in some continuous space and doing things, can we, in the same time, maybe learn to align proteins or learn to align correctly DNA? Or can we use alignment that we know is a good measure of similarity between sequences? Can we use it maybe to supervise tasks? Because this is, we know, I mean, bioinformatics field knows how to recover homology, for example, between sequences by sequence alignment. I must say it's not new. Like there is, for example, this paper published 2 years ago by Bonnie Berger's group on learning. So it's it's a broader thing where they learned embedding. So if you look at the figure here, you see at the top you have protein sequences. Then there is a module called encode. So this literally transforms a sequence of amino acids to a sequence of vectors. And then there are a few things after that, but one of them is about alignment. So they try to to optimize parameters so that when you have 2 sequences after embedding, they can be aligned in a correct way. In that paper, it's not exactly an alignment because there is no constraint. It's not valid alignment. And so we have the question, can we go towards valid alignment and follow what people do in bioinformatics, but in the embedding space? And so this is work in progress. Therefore, I will be quick because there is no publication and I cannot show, I don't have all the results yet. But roughly the idea is to say, suppose you have 2 sequences and you want to learn a module to transform sequences into vectors. So, this is the big topic of my talk, to say we should try to do that. Then a question is, how can you align the 2 sequences once they are embedded as continuous spaces? And so we worked on that and developed algorithms to say, if you have sequences of vectors and not sequences of letters, can you adapt Smith-Waterman, for example, or Goto's algorithms to the continuous setting? And then can you learn to align, meaning that if you know that the true alignment between sequences is given, like if you have some ground truth coming from some experimental data, how do you tune the parameters of the embedding, for example, so that after embedding, your alignment of vectors is good, right? So there are like 2 original things here. One is how do you align continuous objects, and 2, how do you optimize the alignment given a ground truth alignment? And so I will not again go to many details. Suffice it to say that we worked, learning to align is typically how do you optimize the parameters so that a continuous solution to a problem converges to the good solution. It's not trivial because it's not differentiable, etc. But we work particularly on that topic to try to make differentiable discrete values problems. And I will just show some preliminary results. But again, this is not yet, so I cannot give more details. But just show you that there is some hope there. So these plots show on some benchmarks of trying to align sequences, the performance of alignment, so how well you align, and then how well your score is to detect homology. So here there are 2 plots. On the left is for the quality of the alignment, like do you align letters in facing each other in a correct way. And on the right is about homology detection, like is the alignment score large for true homologs and lower for wrong homologs. And here you have 3 curves. The blue, so 2 of them are quite similar. You have 2 blue curves. Sorry for the use of colors. But so the ones which are not very good, let's say, are existing tools. So Blossom, Smith-Waterman with Blossom 62 and some gap penalty, that default alignment. One of them is with optimized parameters. So, but this is existing things. And what you see on the left, for example, sorry, I should say that the performance here is as a function of sequence identity. So what you see is that, and this is well known, that if you have very similar sequences towards the right of the plots, then the performance is high for alignment and homology detection. And when you go to less sequence identity, then it's harder to align and the performance drops significantly. Now, what the red curve, so there is a curve which is higher than the 2 others, is the result of the algorithms I mentioned earlier, the prototype of it. And what we see is that, of course, when the homology, so when percent identity is large, it's easy and it works well. But it seems to work as well quite well when we have remote homologs with very low percent identity. And so this is really the results of learning some embedding, learning some continuous representation of sequences in such a way that after continuous representation, the alignment becomes easier, right? So this is supervised learning, trained on some data set and validated on some independent data set. So it's not final results, but I think it's promising, telling us that even for algorithms which seem fundamentally discrete, like aligning two discrete sequences, going through this continuous representation can be useful. All right, so I'm done. I'm sorry I was a bit long. I hope we will have time for a couple of questions. But just as a conclusion, I think maybe like NLP, we will see BLP, so biological sequence processing, very impacted by all these deep learning ideas. Something crucial here is the idea of continuous representation, so representing a sequence as vectors. There is a lot, and the power of, I did not explicitly say it, but behind that is the power of gradient-based optimizations and end-to-end, what we call end-to-end learning, meaning that once you have represented your sequences in a continuous space, then you can define some objective function and optimize it with gradient descent, which is much harder when you stay in the space of discrete sequences. And so I think it's promising, but as I try to show in this talk, I think there are differences between NLP and BLP, and investigating a bit more these specificities of BLP is probably worth it. There's probably lots of nice research to be done in that front. So thank you for your attention, and if we have time for questions, I'm super happy to take some.
Thank you very much for a very nice presentation. We do have time for a few questions. The first one is by Fanny. So what is the size of reads for classifying species? And could you do classification on genomes or long read sequences instead of short reads?
So in the experiments I showed, we have a mix of simulated and maybe some real data as well, but we worked on short reads of size 200 to 400, I think. Yes, of course, if you want to extend to longer reads or even to full genomes, so maybe the parameters becomes different. It's not metagenomics, but yeah, there is absolutely no problem. And to the opposite, I think it's maybe the future way to store whole genomes would be to embed them as texts. So embed a long genome as a big vector and store the big vector. So yes, I think typically in NLP, so to be super precise, I think there is one constraint in existing NLP model, which is that when they are implemented in such a way that they are good to a maximum size, just in terms of memory, to fit in a GPU, etc. Typical models in NLP take as input a sequence of 1,000 letters, typically. So when you want to process a bigger thing, you need to be a bit smart, either chunk it into pieces or do something. So this requires some specific things, but it's possible to think of embedding longer objects.
Okay. The next question is by Pierre. So what about more complex tasks than metagenomic classification? What about variant detection, assembly? Do you think that BLP could be a game changer in those tasks?
Oh yeah, exactly. I think this has to be investigated. I don't say we will solve everything, but I think it's worth thinking about that. So for maybe the one obvious task that actually we and others work on is for search. Right nowadays, the basic search tools are BLAST or these kind of things. They work by looking at. It's super smart. They work but basically by finding similar sequences, then extending alignment, etc. The obvious way, I work at Google now, so Google knows about search of text, etc. The way it works is done it used to be by searching tokens and words, etc. Nowadays, it's just you have a query, it's represented as a vector, then you have the full internet of document. Each document is a vector, and you do nearest neighbor search in that space, in the continuous space. So I don't say this will work, but that would be completely different from BLAST or BLAST-based search, and it's likely to work to some extent. And for other things like assembly, etc., I think someone has to think about it, but I see no reason why this wouldn't be useful.
Okay, and the last question in the allotted time slot is from Matthias. For accounting for symmetry, what about data augmentation by taking the reverse complement of input sequences? Would it be enough to deal with the reverse complement property of DNA rather than using special architectures?
Yeah, that's a very good point. I think this is an open question, and part in our paper, we barely discuss it, but we discuss it a bit, and there is a previous paper that discusses that. So yes, data augmentation is one way to put not not hard constraints, but to teach the model that it should learn to be, to have coherent predictions. So it works. It can work to some extent, especially in our case where the symmetries are the group of symmetry is small. Like you have one sequence and it's reverse complement, so you just need to show two sequences. But I think this is part of the open questions as well in this field. Like if there is some invariance, should you encode it by design, or should you not encode it and force the model to learn it by data augmentation? It's a bit open if there is benefit in one or the other.
And maybe one last by Laurent. In DLP, the size of the sentence can be very large compared to NLP. How do you deal with that?
So I briefly touched this point in one of the questions before. What we do, for example, is that we work with models that can take as input sequences of 1024 letters, and so when we have a longer protein, in our current prototype, we don't deal with it, but in the future, or even what people do is they typically cut longer sequences into shorter pieces and find ways to merge the representations.
Okay. Thank you. That was all. That's the time is up.
Thank you very much.
Jean-Philippe.